So, as many people know, uh, there have been decades of research done on trying to identify better upfront therapies for patients with peripheral T cell lymphoma. For years, since the National High Priority Study was published in the New England Journal of Medicine, CHOP became a de facto standard of care only because that study, which was focused on patients with aggressive lymphoma, had included a very small number of patients with T cell lymphoma, albeit that number is actually not specified in the patient population description of the trial. But CHOP became a standard. The next biggest step that emerged was retrospective data from the German large cell lymphoma study group published by Norbert Schmidt. And in that study, they did a retrospective analysis that explored all the different regimens they had studied over decades and identified that the addition of etoposide might be associated with some clinical benefit in patients with T cell lymphoma upfront. So in fact, statistically, it really didn't show any improvement in overall survival. There was a suggestion perhaps in some small subpopulations that maybe it did a little better. But because the field is desperate for something, people embraced this retrospective data. And in all probability, if this was in any other malignancy, they would have ignored it. And so for decades since, the field has been oriented toward trying to make advances, and the primary philosophy has been one of CHOP addition studies, right? So if there's a mTOR inhibitor or an antibody, or we'll talk about these trials. But for years and years, these were all single agent trials. And none of them in single arm, uncompared trials demonstrated that they had sufficient promise to warrant moving onto a randomized trial. And in fact, one could reasonably make the statement that most of those trials were associated with significant untoward toxicity. Along comes brentuximab. Brentuximab is an antibody-drug conjugate that targets CD30. CD30 is really one of the defining hallmark features of anaplastic large cell lymphoma. It is sparingly expressed on all other forms of peripheral T cell lymphoma. In fact, 50% of most of the other subtypes have 0 to 5% expression of CD30. But it was really the first drug to come along that was a biologic that targeted a rational component of the T cell biology, and it got very readily approved in anaplastic large cell lymphoma with very high overall response rates, very high complete response rates, because anaplastic large cell lymphoma is that disease defined by 100% expression of CD30. So as part of fulfillment for regulatory agencies in Europe and around the world, there was a requirement to look at randomized clinical trials with that particular drug. And in fact, the trial Echelon 1 was looking at these concepts integrating brentuximab for Hodgkin lymphoma, and Echelon 2 looked at integrating brentuximab with CHOP in patients with peripheral T cell lymphoma. And so the trial is a randomized study of CHOP against C cyclophosphamide, doxorubicin, brentuximab, and prednisone. Now, it's actually the largest trial randomized ever conducted in patients with peripheral T cell lymphoma upfront. And it's a Herculean effort to be able to pull this off. And it basically involved every major key opinion leader in the world. The trial had two important features to think about as you try to put this data into context. The first is the trial required everybody, all patients, to have a 10% or more level of CD30 expression. So the debate and controversy around what is the level of CD30 expression that correlates with clinical activity is really one that's still under massive debate. There's some CTCL data to suggest that maybe the level of CD30 is important, but there's data in other diseases like diffuse large B-cell lymphoma where there was a subset of patients with 30 positive that had some response and patients who were 30 negative who also had some but lesser response. So it is completely unknown what that is. But for the sake of the trial, it required everybody to come in at 10% expression. The other feature of the trial is that it required 75% of the patients accrued to have anaplastic large cell lymphoma. So that could be viewed one of a couple of ways. In fact, that feature of the eligibility criteria was designed in response to regulatory considerations, where in order to secure the approval of brentuximab abroad, there needed to be randomized studies showing benefit in that population. So Echelon 2, in fact, became in part a regulatory study to represent that phase 4 commitment on brentuximab in a randomized way. And so it's for that reason that, in fact, the patient population was stacked toward anaplastic large cell lymphoma. So only 25% of the remaining patients had all the other subtypes of T-cell lymphoma. That included patients with angioimmunoblastic T-cell lymphoma, peripheral T-cell lymphoma, not otherwise specified, and some EDLs and others. There were plenty of subtypes that were allowed in the inclusion criteria for which there were no accruals for those subtypes because they're incredibly rare. The really good news is that Echelon 2 is positive across all the obvious clinical trial metrics that one would look at. And that included both progression-free survival and overall survival. Some have even questioned as to whether there might not be a plateau on the progression-free survival curve and the overall survival curve in the treatment arm. And there are a couple of things to think about. One is the applicability of this to all patients with T-cell lymphoma upfront. And I think it's important to realize what the FDA indication was that was granted to Seattle Genetics for this trial. And the indication actually from the FDA did not specify a certain amount of CD30 on the expression of the cells. Even though the trial said 10%, the FDA kind of said, well, any level of CD30 expression will be taken as sufficient to make you a candidate for the clinical trial. So that's kind of interesting. That's a very generous move probably on the part of the regulatory agency. And so when you're looking at patients with other types of T-cell lymphoma where CD30 may not be prominently expressed at all, the debate amongst practicing clinicians will be, well, is this something I want to do for somebody that has, say, 3% CD30 expression, right? So to help decision makers be confident that it might be a good thing to add brentuximab, the good news is, at least at the first cut, it looks like there's no difference in neuropathy. So the one reason people might have for not choosing brentuximab might be the neuropathy. We've done plenty of studies with brentuximab over the years, and neuropathy is an issue. And in fact, in all these clinical trials, whether it's Echelon 1 or it's Echelon 2, the vincristine or vinblastine is actually omitted to try to reduce that risk of neuropathy in combination with brentuximab. So if you're worried about that factor, at least the first cut of the data says it may not be an issue. To the company's credit, what they did do is they did do a detailed instrument to actually look at neuropathy. That data is not presented here at ASH, and I'm not sure if it's going to be in the publication. The paper looks like it's accepted for publication, and really quite astoundingly, and I believe a testament to the FDA's responsiveness to try to improve options for patients. The application at the FDA was approved in under 2 weeks, and that's remarkably fast. And so I think it implies that there's regulatory support for this, and I think when you see the data and you finally see those curves move, progression-free survival, overall survival, it's definitely a positive step. I think the trial does allow us to ask a lot of other questions. For example, is the observation going to hold up if we just put patients with non-anaplastic large cell lymphoma on these regimens? That's a question that remains to be answered. what is the level of CD30 expression that's really important? Well, for the moment, at least from the community practice point of view, if your patient has CD30 expression and you feel that's the right therapy, you're allowed to go ahead and do it. So I think it's a major advance in a field where we have not had a lot of advances. Now, some might say, well, being addicted to this CHOP-based combination chemotherapy backbone is something we've done for a while. It's taken us 2 decades to identify a trial that finally showed a benefit. So many people, including our group in New York, has suggested maybe we've learned a lot more about the biology of these diseases and that we might be able to leverage many new drugs approved for the disease in recent years, like HDAC inhibitors and pralatrexate, and create novel non-CHOP-based backbones. And in fact, there's going to be data presented at this meeting. One will be an oral presentation from Lorenzo Fauci from my group, who will be sharing phase 1 data of a combination of azacitidine and romidepsin, a pure epigenetic-based therapy, that in a very heavily treated phase 1 patient population is producing overall response rates of 80 to 90% and 100% in patients with angioimmunoblastic T-cell lymphoma. So I actually am the optimist in the field. I actually think this is a good time for patients with T-cell lymphoma, that we have new concepts emerging that are biologically predicated on perhaps replacing chemotherapy, and we finally have insights on new ways to leverage conventional chemotherapies that are FDA approved in a way that we can now demonstrate data-wise is going to help patients long term.